Hello everyone. Today we are going to discuss inflammation into that specific point, that is cell derived mediators. See, while coming to the mediators, or first of all inflammation, as we have already discussed what is inflammation, what could be the reason behind it, that is their causes, etiology. Next is those factors which could be have a direct role into the development of inflammation. We have even gone through the acute inflammation and the chronic inflammation with pertaining factors. Now going ahead with the next point into that inflammation, that is cell derived mediators. As we know that inflammation, what is inflammation? Inflammation is a kind of complex biological response from tissues towards any harmful stimuli, right? So here any type of stimulus or harmful stimulus or the stimuli can lead to have a inflammation. While considering inflammation, or during the process of inflammation, there will be a release of certain chemicals, and they all are mediators. As word indicates, mediators, they mediate the reactions, right? So today we will discuss those cell derived mediators, and myself Dinesh Dangar, and today we are going to discuss cell derived mediators. While considering the mediators, they all are chemical mediators. Further, those chemical mediators can be divided into two: cell derived mediators and the plasma derived mediators, right? So what is mediators? They are basically large number and increasing numbers of those endogenous chemical substances which mediate the process of acute inflammation, and it pertains certain characteristics like that from where exactly it is released, whether they are released from the cells or the plasma. If they are cell derived, then it will be released either from their storage that will be present in cell granules, or they are synthesized in cells. While considering plasma derived mediators, mostly it will be released from liver, and after they release from the liver, these mediators require further activation, so that after that only it can take part into the system or the the process of inflammation. After that, generally all those mediators will be released while it will come into contact with the certain stimuli, right? And those stimuli could be any kind of injurious agent. It could be a dead cells, it could be a damaged tissues, or many more. So here it can be considered as a secondary mediators too, right? Next characteristics of the mediators, like they act on the different targets. They may not have a similar action on the each and individual those cell or the target cell or the receptor site. So they will have a different target for different types of action. While considering their range of actions, right, the range of action of different mediators are increased vascular permeability, vasodilatation, chemotaxis, fever, pain, and can lead to the tissue damage. Here these mediators will have a short lifespan, and once it will be released, that lifespan will be counted. While considering the lifespan, right, after they release, they are generally very rapidly removed from the body by various mechanisms. Like the mechanisms may involve the enzymatic inactivation of it, there will be a presence of certain antioxidants, there will be a certain regulatory proteins, or it may have a even decay itself spontaneously. Okay, so these are the basic characteristics of the those mediators, and the all inflammation or inflammatory reactions involves such kinds of mediators. Okay, so while considering cell mediators or cell derived mediators, or they are also called as a local mediators. Here, as you can see here in diagram, it will be performed mediators, or they are, they will be present in those secretory granules, and these granules are the ultimate store of the, or the storage house of the such kinds of mediators. So here mediators we have histamine, it will be released from mast cell, basophil and the platelets. Next, that is serotonin, it will be released by platelets. Next is lysosomal enzymes, and those lysosomal enzymes will be liberated from the neutrophils and the macrophages. Certain newly synthesized local mediators like prostaglandins, or we used to call it as a PGs or PGs. Next is leukotrienes, platelet activating factors, activated oxygen species, that is free radicals. Next is nitric oxide with their free radicals and the cytokines. So these are all like histamine, serotonin, lysosomal enzyme, prostaglandins, leukotrienes, platelet activating factors, activated oxygen species, that is free radicals, nitric oxide and cytokines. They all comes under cell derived mediators. While considering the systemic mediators or the plasma derived mediators, it will be mostly, it will be released from liver, but further after releasing it may require certain factors, or it may require those activating factors which can have a activating potential. After that only it can work. Here in that plasma derived mediator may involves the factor 12, that is Hedgeman factor. That factor 12 activation, it will done by the kinin system, that is bradykinin, coagulation and the fibrinolysis system. In cases of complement activation, it may require C3A, C5A during anaphylotoxins, and C3B and C5B9, that is from membranatic complex, that is MAC. So now starting with the cell derived mediators. See, in cell derived mediator there are n number of, or we can consider more than 20 cell derived mediators are there. We will go through those important mediators. While considering cell derived mediators, first category into that, that is the vasoactive amines and the peptides. Vasoactive amines include histamine. See here, generally histamines are stored in forms of granules, and those granules will be present within the mast cells. Next is again it will be present in basophils and the platelets. Histamine is released from these cells, that is mast cell, basophils and the platelets, by various agents. While considering stimuli is needed for further degranulation of those mast cells, or degranulation of those mast cell containing vessels or the vesicles or the granules. It is associated particularly with the granules, or in forms of granules. It is obtained from the decarboxylation of histidine. Here histidine is important parameter, right? From histidine, histamine will be released. The degranulation of mast cells takes place in response to various stimuli, such as physical injury. While considering physical injury, like such as trauma, that is the major accidents. Next is due to cold or heat. Here it involves those immune reactions like binding of antibodies towards the or to the mast cells. During that antigen antibody reaction, there will be a degranulation of those mast cell, and once that mast cell will undergo degranulation, it releases histamine. Here the fragments of the complement called anaphylotoxins, those anaphylotoxins like C3A and C5A, can act as any kind of stimuli. Next, neuropeptides like substance P and certain cytokines like interleukin 1 and interleukin 8, which acts as a stimuli. So these are all these 6 kinds of stimuli are there which can degranulate the mast cells and further due to the degranulation of mast cell, there will be a release of degranulation of mast cell can lead to have a release of histamine. Here, while considering histamine releasing factors, it will be released from neutrophils, monocytes and the platelets. Considering the major effects of the histamine, that is the actions of histamine, it causes the dilatation of arterioles and increases the permeability of venules. As during last lecture we have discussed vascular permeability, and due to this vascular permeability, the cells or the the components of the plasma, it may leave the circulation and it may come into the extracellular compartment from artery or vein or from the capillary. So here histamine causes the dilatation of arterioles. Principal mediator of the immediate transient phase of increased vascular permeability. While considering vascular permeability, this could be the immediate transient phase where it involves as a chief mediator or the principal mediator, which can have a increased vascular permeability. Inflammogenic activity of histamine is brought about H1 and partially by H2 receptor. See, histamine has got its own receptors, that is H1 and H2. While considering these two, histamine will have a specific binding affinity towards those H1 receptor and the H2 receptor, and those receptors will be present on each and individual cell and tissues. While considering cell, that cell membrane will have a H1 and H2 receptor. So after release of histamine, that released histamine will go and bind with those specific receptor and can lead to have a certain conformational changes, can have a change in metabolic activity of the cell and the tissues. Next factor, that is serotonin, or the next chemical mediator, or that is from again cell derived mediator in vasoactive amines, that is serotonin, or we can call it as a 5-hydroxytryptamine or 5-HT. Remember, serotonin, or we can call it as a 5-hydroxytryptamine chemically. It has a similar distribution and action to histamine. Their actions will be as same as histamine as well as their distribution. Here serotonin is chemically a 5-hydroxytryptamine, that is 5 capital H capital T, and it will be originated from the platelets. So remember the source. Here serotonin will be released from the platelets. 5-hydroxytryptamine will be distributed inside intestine, that is small intestine, large intestine. It will be present in blood, spleen and the nervous system. The release of serotonin and the histamine from platelets is stimulated when platelet aggregate after contact with the collagen, thrombin or ADP and the antigen antibody complex. Here what it means, while considering serotonin and histamine, as I have stated earlier, these are all mediators. It mediates the reaction, but for that first reaction should be in a progressive condition, or it should be started. Once a reaction is started or initiated, after that only it can take part into it. And further, here there are so many factors or the causes or the agents or the injurious stimulus which can lead to have a certain types of reaction, ultimately leading to release of histamine or the serotonin. So here, while considering serotonin and histamine, that platelet aggregation, and when platelet aggregation can be seen, while during the injury, whenever there will be a, like suppose what we can consider any artery or the arteriole or vein or venules, whenever there will be a bleeding, so that the surrounding surfaces from where exactly the bleeding occur, it may have a platelet aggregation. and that platelet aggregation further releases the serotonin and the histamine. Here, platelet aggregation could act as a factor. Next is even collagen. Next is the thrombin, adenosine diphosphate, and the antigen antibody complexes can release those serotonin and the histamine molecules. Here, platelet aggregation and its subsequent release are also stimulated by the platelet activating factor, that is PAF. It is derived from the mast cells during immunoglobulin E mediated reactions. See, while considering antibodies, we have 5 different types of antibodies, that is IgG, IgA, IgM, IgD, IgE. Right? One more time. IgG, IgA, IgM, IgD, IgE. So there will be 5 types of immunoglobulins, into which specific immunoglobulin E mediated reactions can lead to have a generation of platelet activating factors, which activates platelets to further release serotonin and histamine. Here it will be present inside the those chromaffin cells, which is present in gastrointestinal tract. Again, pyruvate will be present in spleen, nervous tissues, mast cell, as well as the platelets, and their effects will be just like histamine, but it is a less potent mediator of increased vascular permeability and vasodilatation than histamine. Okay, so that is all about serotonin. Next point, that is the bradykinin. Bradykinin is a non-peptide. It is a non-peptide that is obtained from the kallikrein kininogen system. It is a kind of complementary system or complement system, that is kallikrein kininogen system. Here, bradykinin is generated from the tissue damage. Whenever there will be a damage of those tissues, those generated tissues or the damaged tissues can release bradykinin. So while considering bradykinin, even we will discuss bradykinin in detail while studying the plasma mediators, right, where it involves the kinin system. And generally bradykinin acts during the early stage of inflammation, where it may include the smooth muscle contraction. It can lead to have vasodilatation, increased vascular permeability, and the pain. Here, bradykinin is generated from the tissue damage. Next is due to the allergic reactions, viral infection, and due to the other inflammatory events that activates the those proteolytic reactions. Here, it can be divided into 2, that is B1 and the B2 receptor. It will have 2 receptors, just like histamine, H1 and H2 receptor. In cases of histamine, it is divided. It has got 5 receptors, that is H1, H2, H3, H4, and 5. Here, just what we have discussed, that is for only H1 and H2 receptor. As here, in case of a bradykinin, we have a B1 receptor and the B2 receptor, where exactly bradykinin or the released bradykinin will bind. Here, it is also a potent inflammatory mediator, just like similar to histamine and the serotonin. And these receptors increases the vascular permeability and further causes vasodilation, as well as it also increases the prostaglandin synthesis, that is PGs, and it may produce pain locally. And further, the role of bradykinin will be discussed during the next lecture, that is kinin system. Okay, next important chemical mediator, that is from again cell derived, that is substance P. Substance P is an oligopeptide. Again, see, they are the all basically proteins, right? Here in cases of they are oligopeptides, and it is released by nerve endings that has numerous immunomodulatory effects, including the ability to enhance the response of leukocytes towards the or to other inflammatory stimuli. It resembles bradykinin. Their effects will be as same as bradykinin and is further released by the ending of sensory C fibers. It degranulates the mast cells and involved in the pathogenesis of simple responses. See, under the influences of substance P, there will be a degranulation of mast cell. Further, the degranulation involves release of histamine. So here one should understand that even these substances or the released substances or the chemical mediators even can stimulate the other mediators, and it is involved in the pathogenesis of the simple responses. Here, neurogenic inflammatory response of substance P will be mediated through the neurokine receptors. For that, specific neurokine is like histamine. For histamine, H1, H2, H3, H4, H5. Right? Here, in cases of bradykinin, it has got B1 and B2 receptor. For substance P, it can affect on those neurokine receptor. Okay. Next is the arachidonic acid metabolites. See here, I have not included all those chemical mediators. Right? It is not possible that we can cover in such a 30-minute lecture. We can complete all those cell-derived mediators. I have included most important, that is vasoactive from vasoactive amines. I have included histamine and the 5-hydroxytryptamine and the bradykinin and the neuropeptides. Here the next one, that is the arachidonic acid metabolites, and these are all contained, what I am discussing now, that is taken from the Mohan, that is the textbook of pathophysiology, again in that the 7th edition. Here, while coming to the arachidonic acid metabolites, or they are called as a eicosanoids, Here, arachidonic acid metabolites or the eicosanoids are the most potent mediators of inflammation, much more than the oxygen free radicals. So it has got its own function. While considering the arachidonic acid, what is, first of all, what is arachidonic acid? Right, again it is a chemical, as we know that it, because we are discussing it under chemical mediators, right? Again into that cell derived mediators. Basically, arachidonic acid is a fatty acid, and we can call it as a eicosanoic acid. It is eicosanoic acid. Now here, while considering that eicosa or the eicosanoic acid, eicosa, that is a Greek word, it means 20, right? It means 20. So here, while considering the arachidonic acid metabolites, there are certain factors like those it may be released from the complement system or due to the activation of platelet activating factors and the lymphokines. It acts on those inflammatory cells or the cells which leads inflammation. While considering the cell membrane, whenever any cell undergoes damage or lysis or the rupturing, during that cell membrane may have a arachidonic acid. From cell membrane, that arachidonic acid will be liberated or the released. Here arachidonic acid released from the cell membrane with the help of certain kinds of enzymes like phospholipases. Again, it is further then activated to form arachidonic acid metabolites or the eicosanoids by two specific pathways, that is due to the cyclooxygenase pathway or the lipoxygenase pathway. Here in this one specific diagram, I have included all those arachidonic acid metabolites. See here how to understand this, as we know that we are discussing inflammation, in that chemical mediators, how arachidonic acid will be released. Once any cell undergo damage due to the certain kinds of stimuli, it releases arachidonic acid from cell membrane. Now, released arachidonic acid may undergo or it may opt any one of these pathway, or it may may have these two pathways. First, that is cyclooxygenase pathway. Second, that is lipooxygenase pathway. While considering these two, that is those metabolites will be released by cyclooxygenase pathway. See that cyclooxygenase a kind of enzyme that is COX. Here it involves COX, that is cyclooxygenase enzyme. Under the influences of this COX, there will be a release of those prostaglandins, right? So first thing, if arachidonic acid the metabolites that is released from the arachidonic acid, it may opt first pathway, that is cyclooxygenase pathway or the lipooxygenase pathway. In case of cyclooxygenase pathway, there will be a presence of COX enzyme. In cases of lipooxygenase pathway, there will be a presence of LOX pathway or the LOX, that is lipooxygenase. Here, if it follows arachidonic acid follows cyclooxygenase, it releases those prostaglandins, and that prostaglandins can be thromboxane A2, right? Thromboxane A2. Next is interleukin 2, that is I2, interleukins or the interleukin I2. Next is E2, that is actually that E2 means PGE2, that is prostaglandin E2. Here remember one thing, you can refer even other diagrams even if it is need arises. Here it is described in very short manner, right? That E2, F2 and D2, they are the prostaglandins. How we can write it here? Prostaglandins E or the prostaglandin E2, prostaglandin F2 alpha and prostaglandin D2, right? So during this cyclooxygenase pathway, there will be a release of these three specific metabolites. First, that is thromboxane A2. Next, second, I2, that is interleukin 2. Next is prostaglandin E2, prostaglandin F2 alpha and prostaglandin D2. Now we will discuss about LOX pathway, that is lipooxygenase pathway, where there will be a release of leukotrienes. There will be a release of leukotrienes like LTB4, that is leukotriene B4, leukotriene C4, leukotriene D4 and leukotriene E4. So here each pathway will have a separate metabolites. Okay, so here it follows separate metabolites, or it may releases different components at the end of the specific pathway. While considering thromboxane A2 or their effects, it produces the vasoconstriction and the platelet aggregation. See here at lower side artery is shown, it may be a part of artery, arteriole, vein or the venule or from the capillary. We just need to understand the effects. So here in cases of released thromboxane A2, right, that released thromboxane can causes the vasoconstriction of the artery. So there will be a narrowing of the artery, and that narrowing of artery can lead to have a platelet aggregation. So what is platelet aggregation? There will be a, what we can say, that those platelet may form a kind of a layer or it may form a row, right? There will be aggregation or the accumulation. In simple words, we can call it as a accumulation of those platelets. In cases of effects of interleukin, that is I2, even it has got the platelet disaggregation. See here, remember, in thromboxane A2 it produces platelet aggregation, whereas in the cases of prostaglandin into that specific interleukin 2, it may produces platelet disaggregation. So the platelet may undergo suppression, and released interleukin can cause pain. Even E2, that is prostaglandin E2, can lead to have a pain formation. Under the influences of PGE2, PGF2 and PGD2, it may produces vasodilatation. So what it will do? It may undergo or it may lead to have a widening of those arterioles or the venules and the capillaries. See here, while considering vasodilatation and the vasoconstriction, it mostly affects on those arterioles and the venules rather than those major artery and the vein. Next, effects of leukotriene B4. It produces chemotaxis and the neutrophil adhesion. As we have discussed it during the acute inflammation, chemotaxis, this is the process where exactly those chemoattractant factors will be released. Those chemoattractant factors will have a major function, that is to drag out or to stimulate those inflammatory mediators, and it will be further carried up to the site of the inflammation, right? So here it acts as a chemotaxic agent, and it may produces the neutrophil adhesion. So that adhesion, that is what we have discussed during acute inflammation. There will be a margination, and further it will be a rolling, and next is the adaptation and the augmenting. Here, under the influences of LTC4, LTD4 and the LTE4. Actually, these are all that C4, D4 and E4. They are the slow reacting substances of the anaphylaxis, right? And it causes increased vascular permeability and the vasoconstriction. Here, as you can see here in diagram, that increased vascular permeability may lead to have a movement of those substances which may be present inside the lumen that may come outside into the extracellular compartment due to the vasoconstriction. Here, other than these chemical mediators, we can include those lysosomal components like granules of the neutrophils. Here, the granules that is released from the neutrophil even can act as a chemical mediators, that is cell derived mediators. They may be further classified into primary or the secondary neutrophils, or the primary or the secondary components. They are, suppose in cases of primary, they are filled with the azurophil granules. In cases of secondary, they are specific granules and contain alkaline phosphatase, lactoferrin, gelatinase, collagenase, lysozyme, vitamin B12 and the plasminogen activators. In cases of primary granules, they are formed with the myeloperoxidase, next is acid hydrolases, acid phosphatases, next is lysosomes and the defansin. Here next factor could be, or we can consider even platelet activating factor, right? Platelet activating factor is again a kind of cell mediator. In cases of cytokine, even we can include cytokine here like interleukins, right? Again, they are the cytokines or the interleukins into that cytokine, in that interleukins, right? They are basically polypeptides, and it will be produced by the activated lymphocytes, or we can call it as a lymphokines. And those lymphocytes, right, remember WBC cells, white blood cells, right, neutrophil, eosinophil, basophil, lymphocytes and the monocytes, right? Those lymphocytes will release these interleukins, and these are all agents like interleukins. They are self cells, right? They are self cells which produced them or on other cells, right? And in body, mostly more than 200 different cytokines have been described, right? Here in that case, what we need to understand or remember, that is the interleukins. In that, remember interleukin 1, interleukin 6, interleukin 8, interleukin 12 and 17. Actually, I have not mentioned these cytokines here in PPT because you just need to go through the book or the reference book also, right? So I have not discussed here or included the cytokines, right? In that interleukins, remember interleukin 1, 6, 8, 12 and 17, and they may have all effects onto the certain mechanisms or the processes of inflammation. We can even include tumor necrosis factor alpha, TNF alpha, as a chemical mediators, again in that cell derived mediator, because TNF alpha is a mediator of acute inflammation. In that, again TNF can be divided into two, TNF alpha and TNF beta, and it is from, like in cases of TNF alpha, it is formed by the various cells, that is from monocytes, macrophages, mast cells, basophil, eosinophil, beta cells and T cell and the NK cells, that is natural killer cells. Whereas in the cases of TNF beta, it is formed by the B lymphocytes and the T lymphocytes. Here we can include interferon gamma, even other certain chemokines, like even that is MCP, right? Next is we can consider free radicals. It can be of oxygen metabolites or the nitric oxides. So this is all about the cell derived mediators. So what we have discussed till now, the mediators of inflammation. What is those mediators of inflammation? The characteristics of mediators of inflammation. Next is cell derived mediators. In that we have already discussed vasoactive amines, in that histamine, 5-hydroxytryptamine, neuropeptides, tumor necrosis factor alpha. Next is platelet activating factors. Then we have moved to those arachidonic acid metabolites, that is eicosanoids, into the two specific pathway, cyclooxygenase pathway and the lipoxygenase pathway. In cases of cyclooxygenase, we include thromboxane, interleukins. Next is prostaglandins E2, F2 alpha and the D2. In cases of lipoxygenase pathway, where it includes those leukotrienes like leukotriene B4, C4, D4 and the E4. So this is all about cell derived mediators. Next hour we will have a discussion on plasma derived mediators. Thank you very much.